for the case of anencephaly, we expect, or we can expect that the forebrain is not going to develop normally since the anterior neuropore did not close properly. So the forebrain prosencephalon will be in constant exposure to amniotic fluid, so we don't expect it to develop normally. For pregnant humans, for pregnant women, okay, to avoid neural tube defects, clinicians will normally prescribe an intake of supplementary folic acid. So, it's been shown that increased folic acid supplementation during pregnancy prevents the likelihood of developing neural tube defects like spina bifida or anencephaly to the newborn. Let's further dive into the process of primary neurulation by examining a few hypotheses on how uh, the three groups of cells, the three groups of tissues, are actually delineated from one another as the neural tube develops and sinks into the coelomic cavity. So we expect the neural inducting components and factors to just function normally as they do, right? On the macroscopic or cellular level, right? We're seeing that um, a key factor in the delineation of the three layers or the three tissues, the three groups of tissues or three groups of cells that develop after neurulation is the presence and absence of CAMs, cell adhesion molecules. Alright, so on this diagram, we are seeing the delineation of the neural plate that will become neural tube from non-neural ectoderm. So the initial stage, neural plate shaping, alright, and bending is represented by this picture. So the blue colored or the blue labeled part is competent neural plate responding to inductive signal, while the orange colored part on the sides of the responding neural plate are non-neural ectoderm. So we presume them as cutaneous ectoderm. Right? So few experiments detailed the presence of cell adhesion molecules, of calcium dependent cell adhesion molecules on these tissues. It's been shown that in neural plate or responding neural ectoderm, special kind of cell adhesion molecule known as LCAM, uvomurelin, or E-cadherin, is present originally, okay, in neural plates. So LCAM rather, is present. No. Later on, this LCAM or E-cadherin, which is present in the original neural plate, will be gone. Alright, so it will be replaced by another type of cell adhesion molecule known as neural cell adhesion molecule or NCAM. Alongside with it, N-cadherin is also going to be present. So your neural plate loses E-cadherin and gains N-cadherin and NCAM. So as it gains N-cadherin and NCAM as cell adhesion molecules, it will no longer associate itself with the other neural plate cells that are not really going to become contributory to the formation of the neural tube. So you expect that the presence of the cell adhesion molecules actually allow the groups of cells to detach from one another. And to prove this, right, some experiments were done to check if N-cadherin, E-cadherin is really an important factor. So in diagram B, we're seeing that if our neural plate will be allowed to possess both NCAM, N-cadherin, and LCAM, E-cadherin, neural folding, proper closure of the groove forming the neural tube will fail.